Hello and welcome to Nature Knowledge. This is a speaker series with experts sharing scientific knowledge on current issues affecting nature in Florida. I'm your host, Dr. Shelly Johnson, State Specialized Agent in Natural Resources with University of Florida IFAS Extension. Thanks for joining us today.
So let's go ahead and get started with our topic for today. Florida red tide is an issue that tends to come up every year around this time, so we thought it would be a good, appropriate time to discuss it. And I've invited back Dr. Lisa Krimsky, who actually was with us in April to talk about blue-green algae. So this is, we went really well, and so I'm excited to have her back. And she's the Water Resources Regional Specialized Agent with UF IFAS and Florida Sea Grant. So I will turn it over to Lisa, and I will stop sharing my screen so that she can share hers.
Great. Thank you, everyone. I'm glad to be back to the this webinar series. I'm excited to be talking to you all today. Let me pull up my presentation. And is everyone seeing it? Okay, perfect. So because of the season, I was asked to talk about Florida red tide, and this is going to be sort of an introductory presentation, but I welcome questions, and if I can't answer anything now, I'll definitely get back with you. But I also, before we get started, in addition to the poll at the end, I'm going to be trying something new today as a presenter. I'm going to be asking you some questions throughout using an interactive polling platform. So at discrete points, and it'll be very obvious, I'm going to ask you either to use your phone or to pull up your web browser if you're sitting in front of a computer and work with me to answer some questions, and we'll see how it goes. So because we're talking about red tide, which is an algae, I wanted to quickly go over what algae are to kind of frame this presentation. So algae are an extremely diverse group of photosynthetic organisms. They are plant-like, but they lack a lot of the major plant structures, such as leaves and roots that we normally think of. They are primarily aquatic, but they are found essentially anywhere on Earth that has sunlight, again, because they are photosynthetic. So they're found in both marine and fresh waters. They do include cyanobacteria, which are not true algae, but they're sort of fall within that larger umbrella term. They also include seaweeds, which are larger macroalgae, multicellular structures that tend to be attached to the bottom of aquatic habitats. And you can also be found in terrestrial places as well. So for those of you who may or may not know, lichen is actually a combination of fungi and algae. So algae really are pervasive, they're everywhere, and they are extraordinarily important to Earth as we know it. So the cyanobacteria that I mentioned before, they are responsible for the oxygen that we have on planet Earth. They are the reason why we are all able to live here. And even today, phytoplankton are responsible for about 50% of the oxygen in our atmosphere. So again, extraordinarily important in terms of oxygen production in our atmosphere. They're also important for carbon sequestration, again, for that photosynthesis. They take carbon out of the atmosphere and produce oxygen, and they are responsible for sequestering about 1/3 of the carbon on our planet. So again, extremely important. Because of all of this production, they are the primary producers in aquatic food webs. They form the base of those food webs and are responsible for all of the aquatic systems as we know it. They're also really important for a lot of other biogeochemical systems. They take nutrients and transform a lot of other, such as metals and other things in our system. So they're really important, not just for atmosphere and carbon dioxide, but in terms of nutrient removal and sequestering a lot and transforming a lot of other metals. And then for those seaweeds that we talk about that are structured to the bottom of aquatic habitats, they actually can help to stabilize the sediment, preventing erosion. So again, I want to note algae tend to get kind of a bad rap. They're extraordinarily important for life as we know it on planet Earth, and only about 1% of the algae are known to produce toxins. So when we talk about algae, we tend to classify them into 5 major categories. So that's green algae, diatoms, brown algae, red algae, dinoflagellates, and then cyanobacteria, which again are kind of that 6th, not real algae, but falls within that greater umbrella. The species that we're going to be talking about today, Crania brevis, red tide, is a dinoflagellate. And the first thing that you should notice is that these common names, these major classifications, are extraordinarily confusing with what we tend to call algae blooms. So Crania brevis, red tide, is a dinoflagellate. It's not a red algae. And cyanobacteria, this blue-green algae, are not green algae. And so it tends to get really complicated and really confusing when we talk about these species and algal blooms and understanding the complexities associated with algae and bloom dynamics. So what's important to note today is that Crania brevis is called colloquially or casually red tide. That's because when you have large concentrations of these cells, when there is a bloom, it can turn the color of the water red. Not always. Sometimes it's brown, sometimes it's yellowish. They are dinoflagellates. They are not red algae. And they are also not the only red tides that we have. So there are a number of harmful algae forming species that also are called red tide. In Florida, the red tide species is Crania brevis, but if you're in, say, New England or the Gulf of Maine, it's a totally different red tide bloom species. If you're in California, it's a totally different red tide bloom species. And so the whole point of this slide is just to say that we try to simplify things when we're talking about science and communicating about science, but we oftentimes complicate things unintentionally. And so keep in mind that today we're going to be talking about Crania brevis red tide, which is a dinoflagellate. So what are algae blooms? Algae is the rapid and substantial increase in algal biomass in an aquatic system. And a harmful algal bloom is when you have that rapid and substantial increase in biomass, but that you also have toxic or harmful effects on human health or aquatic ecosystems. And so again, referring back to that previous slide, not all blooms are the same. And just because you have a bloom of a species that can be toxic doesn't mean that that bloom necessarily will be toxic. This is especially true with a lot of the cyanobacteria species, not necessarily Crania brevis that we're talking about today. So what causes algae blooms? The picture on the right shows all of a variety of different nutrient pathways that can lead to algae blooms and hypoxia. What's important to note is that nutrients, while essential for algae because they're plants and they need nutrients to grow, they are not necessarily the sole instigator of an algae bloom. So for example, there are a variety of different harmful algal bloom forming species, such as the Crania brevis red tide, that do not bloom in response to excess nutrients by humans. And so there are a lot of other chemical and physical associated factors that will trigger this bloom, not necessarily human-induced nutrients. And so other issues might be water clarity, circulation patterns, hydrology, climate and weather, and then biological community interactions. And so HABs are natural, especially for those bloom-forming species that don't bloom in response to anthropogenic excess nutrients. But when you have persistent blooms, that indicates that there's a likely ecosystem imbalance. So this is just to show that we are not alone in the state of Florida. We have harmful algal blooms in every of our 50 states, Although, if you take a look at that map and all of those pretty colors, we do have the most diversity of harmful algal bloom forming species in the state of Florida. This map shows the distribution by the related syndromes. And so those are sort of the negative associated symptoms associated with the presence of a harmful algal bloom. In this case, the Crania brevis red tide is indicated by that large purple oblong shape because the toxins associated with Crania brevis are a neurotoxin, and the syndrome is neurotoxic shellfish poisoning. So I don't want you to get sort of confused or wrapped up in all of these HAB-related syndromes. The purpose of this map is just to show that Florida has a lot of different HAB-forming species, and that's because we have a lot of water in the state. We have a lot of warm temperatures, a lot of sunlight, all of those key triggers that are necessary for increasing algae growth. This is kind of just an overview of HABs, just to one more time sort of hit it home that we have a diversity of different bloom forming species in the state. They really, if there is water, there is a possibility, whether it be fresh water or marine water, there is a possibility that a bloom can occur there, and that the diversity of species causes complexities in terms of management because not all blooms are the same. Okay, so now we talked about, in general, sort of algae blooms and harmful algae blooms. I'm going to focus now on Crania brevis red tide. So Crania brevis is, as we mentioned, it's a marine harmful algal bloom, meaning that it persists in salt water, and it is a dinoflagellate, which dinoflagellates have sort of a flagella which allows it to swim. And Crania brevis is actually one of 6 species of Crania in the Gulf of Mexico. And it is this unique swimming pattern that allows scientists to be able to determine whether it's Crania brevis or one of the other species of Crania that we have. It is also the dominant species of Crania in the Gulf of Mexico. As mentioned, it was first confirmed in Florida in 1844. But there is evidence to suggest, going all the way back to the 1500s, that red tide blooms might have persisted. The Spanish, when they came over to Florida, there are notes and records indicating red colored water and fish kills and dead marine birds. So again, linked to or associated with potential for red tide blooms, although it was never, obviously couldn't be confirmed. Crania brevis is prevalent in the Gulf of Mexico, and it's always there. It's always found in background concentrations in the water column, but it tends to bloom almost annually in late summer, early fall. That's why here in late August, I'm talking with you about red tides, because 'tis the season. And it's considered a harmful algal bloom, again, because of the harm that it causes with the toxins associated with it. The other thing that's important to note about Crania brevis is that it's a slow growing organism. And this is important when we talk about when and why we may or may not have a bloom. So when do red tide blooms occur? How do they start, or what we scientists call initiation? So a bloom tends to start and begin in deeper waters, in offshore waters, along in the Gulf of Mexico, along the West Florida shelf. And it's important to note that this area where blooms normally start are actually low in nutrients. And so you can wonder, okay, well, red tides, even though they're an algae and they grow offshore, they still need nutrients to grow. So where do they get those nutrients from? Well, there tends to be sort of an association between the start of a red tide bloom and another type of algae bloom that also occurs in the Gulf of Mexico. You might have heard of the cyanobacteria trichodesmium. This is a cyanobacteria, again, that's in the Gulf of Mexico, and it has a unique advantage of being able to take nitrogen out of the atmosphere and convert it into a biologically usable form. So that's something that's unique to some species of cyanobacteria. Trichodesmium is one of those species, and it also requires these micronutrients, iron being one of them. And so trichodesmium blooms tend to be associated with Saharan dust clouds. They bring iron. Trichodesmium takes nitrogen out of the atmosphere, converts it into a biologically available form, and Crania brevis comes in and says, ooh, I'll have me some of that. Thank you very much, trichodesmium. And it starts to bloom. So it tends to often happen again at depths and offshore at the zone between Tampa Bay and Charlotte Harbor. So there can be a bloom. You can have large concentrations of Crania brevis offshore, and we don't necessarily know about it because blooms really only become harmful or nuisance to us once they come inshore, which is where we are. So they get transported and tend to grow through ocean circulation patterns. This is showing a picture of upwelling, which is when winds at the surface blow offshore and actually push the water away from the coast. That allows deeper water from offshore to move in and sort of fill that gap left by the winds that have sort of left this area open for water. Remember, I said that Crania brevis blooms start in the deeper offshore waters, about 10 to 40 meters out. And so when those upwellings occur, it brings with it that cold water transports the Crania brevis cells. And that's how blooms get transported inshore and when they really start to become harmful to us. Whether or not a bloom is going to persist is also going to be dependent on a lot of ocean circulation and wind patterns. The loop current in the Gulf of Mexico being one of the most important circulation factors. So the size and the intensity of the loop current is really going to determine whether or not we have a big bloom or whether or not it gets transported onshore. So the loop current will help to bring up, again, a lot of those nutrients from the bottom. But if you have a really strong, intense loop current, it's going to bring up a lot of nutrients. And if you recall, one of the biological features of the Crania brevis is that it's slow growing. So if you have a strong loop current, if there are a lot of nutrients readily available, it will actually get outcompeted by another type of algae, most likely a diatom. If it's fairly weak, that's when Crania brevis will utilize those nutrients and start to outcompete and become persistent and allowed to bloom. You also need the movement of the loop current near shore to be able, again, to transport it onshore, coupled with that upwelling circulation. So once it's onshore, that's when those human sources of nutrients really become part of the factor. So Crania brevis is really flexible in where it can live. It has a very broad salinity and temperature tolerance. Generally, it can grow at about temperatures higher than 24 degrees Celsius and in salinities between sort of 20 and between 22 and 25 parts per thousand. So it likes marine warm waters, but it does have a very broad range. It also can be hindered by light intensity. It needs strong light in order to grow. But it's, in addition to having sort of this broad tolerance of salinity and temperature, it's capable of using upwards, I think it's now 13 different sources of nutrients for growth. And so once it has started and once it's started to grow, sort of the opportunities are endless for this organism. And that's why red tide blooms can get so big and cause so much havoc to our Florida coastal waters. So some of these sources of nutrients we already discussed, the Saharan dust triggering the trichodesmium blooms. You can have sediments buried in the nutrients buried in the sediments that flux out. You actually have nutrients from the atmosphere. So when it rains, atmospheric deposition, that's a source of nutrients. You have those human-derived nutrients from fertilizers and septic systems and sewage leaks. You also, one of the real sort of, I'm not gonna say clever, I don't wanna anthropomorphize an algae, but one of the tricky things associated with the red tide algae bloom is that it can get a lot of sources of nutrients from the fish kills itself. So once a bloom occurs, once it's near shore, it can cause fish kills because of the brevitoxins, and then the nutrients released from those decaying fish will actually cause sort of this new source of nutrients. So it becomes sort of this cycle of nutrients that become available. There's potential other sources of nutrients. In 2018, we had the two blooms, the cyanobacteria blooms and the red tide bloom happening at the same time. And there was a question as to whether or not nutrients associated with the cyanobacteria blooms could be fueling these red tides also. That remains to be seen. We do know that estuary discharge coming out of the Caloosahatchee is a source of nutrients for red tide, but it's a relatively small source of nutrients compared to the other nutrient pools. One of the other things that we don't know about in sort of this whole life cycle of a bloom is what is actually responsible for terminating a bloom. That's a scientific question that we don't know yet, and is one of the top priorities to figure out is ultimately what causes a bloom to die out. All right, so this is the first question time. So I'm going to ask you, if you're at your computer and can open up a browser, to go to menti.com and enter the 7-digit code that you see on the screen. If you have a phone, you can just take a picture of that QR code. I'm going to give you a few minutes to, not a few minutes, a few seconds to do that, and then I'm going to switch the screen over. If I can figure out how. Okay. So I'm going to switch the screen over. You should still be able to see the code. So for those of you who are still trying to figure out Menti, again, go to menti.com, use the code. And what I'm asking you is, when you are looking for information about red tides, what's most important to you? What is sort of the primary information that you want when you're searching for red tide information? And so what this is doing, this is actually taking all of your responses and it's creating a word cloud. So causes is a biggie. Everyone wants to know what causes the red tide. I see toxins and health going up there. I see toxicity, location. All right, keep going. I've got about a minute left. I'm gonna just keep letting you put your information in there. All right. A minute's a very long time. We're gonna close this before a minute. I'm gonna give you 10 more seconds. So type fast for those of you who are still working on it. But again, the biggies seem to be causes and toxins, severity, health. Okay, I'm gonna flip back over. That worked pretty well. Thanks, guys. That was a fun little experiment. Okay, so again, a lot of you talked about toxicity, health. So then I kind of anticipated that. And so the next portion of the talk is going to focus on Brevitoxin, which is a neurotoxin in the dinoflagellate Crania brevis. And it can have impacts both on humans as well as ecosystem health. Most of us are familiar with the fish kills images from previous blooms, which are absolutely devastating. And that's because of this brevitoxin associated with the red tide organism. So the brevitoxin actually can become aerosolized, which means that once the cell lyses or splits open, the toxin becomes available and can be moved through the air, either captured, most likely captured on water droplets. And there have been studies to show that these water droplets with the attached aerosolized brevitoxins can be transported over 1 to 2 miles over land. The toxin itself can also adsorb onto biological surfaces, so it can be persistent in food webs after blooms subside. So there have been seagrasses that continue to have toxins associated with it. I saw a couple of you put sea turtles and manatees, and that's likely, again, even though a bloom may have persisted, some of those food sources might still have toxins associated with it. And so there is sort of this feedback loop. And then it's also directly consumed by filter feeders. The toxin can be ingrained within the tissues of these filter feeders, which becomes, again, a problem as it bioaccumulates into the food chain. So for humans, the major sources of exposure, because it can be aerosolized, is that airborne inhalation. For most people, the inhalation really is not too much of a problem. You might get sort of an itchy cough as soon as you remove yourself from the situation and go into air conditioning. It will likely subside pretty quickly. It can have serious complications to those with respiratory ailments, those with asthma, for example. And so it is recommended that if red tide is in an area and you do have respiratory issues, that you do not go to the beach during those times. It can cause skin irritation and burning eyes. A lot of questions that we get is, can you swim at the beach when there's a red tide? You can swim at the beach, but if you know you have sensitive skin, don't do it. It's just not worth it. Go somewhere else. And also, obviously, don't ever swim anywhere where there's a fish kill, because then you're just bringing in a whole lot of other factors to complicate the situation. It also can be ingested. And again, as I mentioned, it can be accumulated through filter feeders into the tissues of organisms. And so the consumption of contaminated shellfish is the primary source of ingestion. And because of this, we have a really highly regulatory process in the state of Florida for shellfish harvesting beds. And it's monitored extraordinarily closely, and it's closed anytime you have red tide concentrations at 5,000 cells per liter or higher. And because of that, and because it's been so successful and it's so strict, we actually have no known sicknesses or deaths associated with Crania brevis neurotoxic shellfish poisoning in commercially harvested shellfish in the state of Florida. We do have some illnesses associated with shellfish, but those have all been recreationally caught shellfish. So people who are harvesting when they shouldn't be harvesting in areas that have Crania brevis, and they may not know it. But there have been no commercially known illnesses. And although not pleasant, there are no known deaths associated with the neurotoxic shellfish poisoning from Crania brevis. But it is odorless, it is tasteless, and it cannot be, the toxin cannot be degraded with either heat, cold, or acids. So all of the amazing ways that we enjoy eating our seafood, it doesn't get degraded. One of the questions that we get asked all the time is finfish. Finfish are safe to eat. Do not eat the viscera. That means the internal organs of the fish, because that's where the toxins get accumulated. But the fillets of fish, the toxins are not concentrated in the tissues. So in addition to the environmental impacts and the potential health impacts, we know we have a lot of economic impacts, cleanup costs associated with the removal of all of the dead fish, loss of tourism, business, as well as potential loss of jobs, fishery closures last year during that persistent red tide bloom. The shellfish harvesting, the aquaculture and shellfish regions was closed for over a year. So it was essentially an entire year where a commercial fishery just had no income associated with it. Your human health costs associated with going into hospitals or loss of work days, and then coastal property values. But the good thing is that we have a lot of tools at our hands that can mitigate all of these impacts, including management, monitoring. As I said, we do have a really robust and successful monitoring program for the shellfish industry, outreach, and communication tools. So current conditions, we are in August. We are at that time of year where we're likely to have a red tide if there's going to be one. And I am pleased to say that this is a red tide current status daily sample map from yesterday. You can see that there is no or background levels of Crania brevis throughout the majority of the Gulf of Mexico. There is one site that has a low concentration, which you can see highlighted there in Stump Pass. But again, a low concentration means that unless you are prone to having respiratory ailments, it is primarily safe for you to go to that area. Fish kills are possible, but unlikely. But those of you might have remembered from way back in June, all of those beautiful sunsets and sunrises that we were having and all that colorful skies was because of the huge Saharan dust clouds that came through, spreading over 2,000 miles across the Atlantic. And with that Saharan dust brought iron. And remember, what is that a trigger for? Oops, stop playing. That's a trigger for trichodesmium blooms, that cyanobacteria bloom that's able to take nitrogen out of the atmosphere and convert it to a biologically usable form. We do have trichodesmium blooms in the Gulf of Mexico right now. This is a picture from FWC of a concurrent trichodesmium bloom and a red tide. This is not a current picture, so don't worry. We don't have red tide in the Gulf of Mexico as we speak, but we do have trichodesmium blooms. And you can tell the difference. The gray, sort of sawdusty looking bloom is the trichodesmium. So what does that mean? We don't know, but it does mean that there's the potential nutrient source, at least in those nutrient poor open waters, for Crania brevis to persist. But again, there's a lot of other oceanographic, physical scenarios that sort of have to align in order for a Crania brevis bloom to occur. So we're kind of at a wait and see situation right now. But I noticed in some of the comments when you all registered, someone said that they still have nightmares about the 2018 bloom. So we do have to acknowledge that. The 2017, 2018, 2019 bloom was a massive bloom. It was first noticed in October. You can see October 2017 on the left-hand side here. And at its peak, 1 year later, it had hit all three coasts in the state of Florida, which is kind of unprecedented. I think the last time a red tide bloom had hit the East Coast was 7 years prior to that. So it's not something that happens on a regular occurrence. It was since the state of Florida had started monitoring red tide blooms in the 1940s, it's indicated to be one of the 5th longest bloom on record. But it's important to note that because of the way monitoring occurs and changes of the monitoring plan and program over time, you're not able to compare bloom seasons to bloom seasons. So while we can say that, yes, it was an extremely long bloom, we can't compare intensity to previous blooms. So we can't say this was a worse bloom, although we know that it was a long bloom. So what happened? Well, folks at USF who do work a lot in terms of monitoring red tide blooms, they actually figured out that it was 2 blooms. So you had the bloom that started in October 2017, and it kind of lingered and hung around. And then you had the storm, which brought the red tide into the panhandle, in addition to a 2018 bloom, which started offshore at the same time that we had the 2017 persistent bloom. So it was actually two different blooms sort of coinciding at the same time. And then with the hurricane, you had the transport of that bloom to other areas in the state, impacting both the panhandle and the East Coast. So it's kind of like just all of these weather, climatic, oceanographic scenarios that just kind of brought out, you know, the perfect storm. Question time. You guys are all pros at this now, so I'm going to switch over again and ask you the next question. So you just told me what's important to you. Now I want to know, who do you go to for that information? Where do you get your red tide information from? All right, seeing a lot of the usual suspects. All right, I'm gonna give you maybe 10 to 15 more seconds. All right, perfect. I'm gonna switch back. Okay, so where do you go to? Well, let's talk about some of those resources and management for Florida red tides. These are the players at that large scale. I'm not going into the details in terms of individual universities. I just showed you information from USF. We obviously are UF IFAS. I noticed FGCU being local, they were mentioned, FIU, as well as a number of news sources. Those are all important members of sort of the communication and outreach community. From an agency perspective, these are kind of the main players. Moat Marine Lab is up there because they actually have a joint partnership with FWC, Florida Wildlife Research Institute, for managing red tides. We have the Florida Department of Agriculture and Consumer Services. They are responsible for the shellfish harvesting beds. So in making sure that they're monitoring and that the commercial shellfish industry is healthy for us. In terms of health, human health, it's the Florida Department of Health. NOAA has a lot of fantastic resources that are available that I'm gonna show you some of them. And then Florida Department of Environmental Protection, well, they work primarily on the freshwater side of things, and FWC works primarily on saltwater marine HABs. There's a huge concerted effort at the state level now to get all of the agencies working together. And so they are becoming partners in harmful algal bloom communication. So where do you find the data? The most recognized resource is FWC, the Florida Fish and Wildlife. Their research, the Florida Wildlife Research Institute, runs the statewide monitoring program for Crania brevis. So you can go to the website listed there. If you click on the daily sample map, that's that interactive GIS map that I showed you previously, where you can actually click on the individual locations. Or if you go under current conditions and you click on report and map, that'll give you the static image showing you the locations just in a picture format that you can see here on the right hand side. And that gets updated weekly, whereas the daily sample map, as the name indicates, gets updated daily. NOAA has two products that are available. On the right hand side is the monitoring system. So this is a satellite system that they have. They also have satellite imagery for inland Floridas, particularly Lake Okeechobee and the coastal estuaries. This is an image from August 10th. It was the most recent image I could find where cloud cover wasn't covering the entirety of the coastline. And what you can see here is that, oh, there looks like chlorophyll. There's a presence of something. That chlorophyll signature is actually going to be those trichodesmium blooms, because we don't have any active red tide there now. So it's an extraordinarily useful tool. It's critical in determining where specifically harmful algal blooms are, but you can't get sort of species specific when you're looking at it as a standalone product. They also have a HAB bulletin. Oop, I apologize for that typo. This is a sort of a respiratory conditions report. This is from last year because, again, we don't have any red tide now, so the entire map of Florida is blue. But what they do is they go in and they comparing wind and concentrations and all sorts of data, as they're able to show you, they segment the state into areas and sort of color code respiratory irritation levels. And so if you just Google NOAA HAB bulletin, you can get that. USF has a big program in terms of again, as I mentioned, monitoring Crania brevis in ocean circulation. And they have a forecasting tool which kind of allows you to predict where it's likely to go. And so again, these are not recent images because the images are blank for today. But it shows you based off of, again, wind, ocean circulation patterns, Crania cell concentrations and locations, where it's likely to be forecasted in the future using a color-coded system. Oops. So in 2019, when the governor came on board, one of his first things that he did was he started the blue-green algae and the harmful algal bloom task forces. And the harmful algal bloom task force was tasked within the first year of work to address specifically red tide, although it's not the only harmful algal bloom in the state. They came up with a consensus document for recommendations regarding red tide blooms in Florida. And they had identified information gaps and priority needs for public health, communication, management, and response, as well as research. And for this first year, they prioritized communication as well as real-time technologies for detecting Crania brevis blooms and Brevitoxins. And so the state of Florida supported a call for grants, and they funded 2 projects. I want to let you know that you all just helped me in this project. I received funding to do the communications portion, and through your Mentimeter responses, you just helped me sort of assess the first level of surveys as to where people get their information from and what's most important to you. So thank you for contributing to Red Tide Science and Communication. We also want to reduce favorable bloom conditions. So again, I hope you recognize that red tides start in low nutrient areas offshore that are removed from any human sources of nutrients. So while humans can't play a role in the initiation or the start of a bloom, once that bloom gets transported near shore, the human sources of nutrients absolutely can play a role in fueling or sustaining those blooms. And that's where we really have a role to play in terms of mitigating those sources of nutrients so that we can at least help to reduce the intensity or duration of those blooms. And you as individuals can make informed decisions. So you can make decisions about sort of how you manage for nutrients, how you work to address in your local communities nutrient management solutions. You can make really important decisions for yourself as to whether or not you go to a beach on any given day. If there's a red tide in the area, make yourself an informed citizen. Know where the red tide is and where it isn't, because very rarely is red tide, it's an extraordinarily patchy bloom. And so very rarely is it going to impact all areas of part of the state. And so you can do your part to know where it is and where it isn't and still help and support local economies while keeping yourself safe. And be part of the solution. There are citizen science opportunities for red tide. HabScope is a citizen science project where individuals, I know he's covered, but it's an individual who goes out and collects water samples, then puts those water samples in a microscope that's linked to an iPhone that has a specific app in it. That iPhone uploads the data. It takes video imagery of the water and is able to detect the Crania brevis cells and concentrations by the swimming patterns that I showed you earlier. And then that is uploaded and analyzed and able to give near real-time forecasting tools for local beaches. So this is something that NOAA and GCUS, who are partners in this, are really trying to get to the local beaches to increase awareness and get more robust coverage for real-time Crania brevis concentrations to ensure that people can go to the beach and stay healthy at the same time. So with that, I'd like to thank you all for joining us today, and I will take any questions.
So there are a few questions in the chat. Some of them you may have answered already, but.
Okay, do you want me to scroll through?
Yeah, if you want. And then Haley can let us know if there's any questions from the Facebook page.
Okay, so the first question is about the Saharan dust being an influencer. And as I mentioned, Saharan dust sort of non-directly through being supportive of the trichodesmium blooms, which then thereby supply a source of nutrients for Crania brevis. Long-term health impacts of brevitoxins, I'm going to be honest and say we don't know. The long-term health impacts is one of the biggest gaps in our public health knowledge and is a huge focus for scientists right now and the research community and the management community, because we recognize that there's a big gap in that. Yes, there is a recent Biscayne Bay fish kill that's related to low oxygen. To my knowledge, it has not been linked to an algae bloom. I don't believe there's any algae in Biscayne Bay right now or any algae blooming. Let me rephrase that. I was understood that it was high temperatures were causing the low oxygen, and then lack of circulation was resulted in the fish kill. Can radiation be used on shellfish to kill the toxin? I honestly don't know the answer to that, but I can look into it and get back to you. Thank you for sharing the current red tide status. How are blooms affected by warmer water temperatures? So most algae blooms prefer warm water temperatures, and Crania brevis, as I mentioned before, is pretty ecologically flexible. It has a really wide range of temperature, and I think like it likes water temperatures into like the high 30s. So I don't think for Crania brevis, warm water temperatures is a limiting factor. Yes, being a dinoflagellate, they can move up and down the water column and take advantage of different nutrients. So yes, in addition to their swimming, they're able to sort of vertically change where they are in the water column and again, take advantage of not only of nutrients, but of light. Remember, low light can be a limiting factor for them. So that also gives them an added boost. Human nutrients play less of a role in other sources in initiating a bloom, but once a bloom has formed and is brought on shore, human nutrients definitely play a role. And so in terms of controlling it, so there's no way to prevent a bloom. In terms of controlling it, so once a bloom has started, again, that's another area of research that's become a major focus right now. They're looking at a lot of different options for controlling blooms once they've already started. One of the biggest is clay flocculation. So it takes sort of these really fine clay particles that bind to the cells and bring them down to the bottom without releasing the toxin. It's been used in other blooms in other countries. And I believe there's trial testing going on in the Florida. The issue is that red tide can be so widespread that financially and feasibility of doing it at a large scale kind of remains to be seen. There's other, once it gets into enclosed sort of canals or nearshore waterways, there are other options like ozonation and other different types of management strategies that are currently being evaluated. Again, that's another major focus. And that's one of the main things that Mote Marine Lab is actually working in concert with FWC right now on, is managing grant funding for technologies for control.
Okay, so we have about 5 minutes left to keep answering questions. I am going to launch the poll really quick. And you can keep answering questions while this is up for folks to answer.
Okay. Stuart, the percentages of the 12 different nutrient sources, I don't know them offhand, but there is actually a paper that shows sort of the relative contribution of the different nutrient sources. So I will send that your way. I assume that email addresses were given during registration. Where can you volunteer to do local water sampling? I am going to put in the chat. I'm going to put in my colleague, her email address, Betty Stogler. She can get you in touch with the folks at GCOOS who run the HabScope Citizen Science Monitoring Program. So thanks for being part of the solution. It's hopping around. Okay, summer fertilizer ban in an attempt to reduce nutrients. However, iron is recommended as a substitute. Is this a wise choice? Probably, because the concentrations of iron that are coming from land-based applications, again, are so small, and the trichodesmium is offshore, that land-based application of fertilizer is an iron-based fertilizer is really not likely to impact that offshore trichodesmium bloom species, cyanobacteria. Is there evidence that red tide events are now or more or less frequent or severe than in the past? So this is something that gets brought up a lot. For red tide specifically, we have no way to compare because monitoring of red tide, as with most monitoring, ebbs and flows. The finances to support monitoring ebb and flow with the times. So the monitoring program has changed. The intensity and the resources of the monitoring program have changed over time. So we can't compare red tide data from the past with red tide data now. It's like comparing apples to oranges. That being said, on a global scale and even on a national scale, there is evidence to suggest that harmful algal blooms in general are increasing in frequency. Okay, Tracy from Facebook. When I talk about nutrient dumping, am I considering the waste from Mosaic? Along with the blooms, I'm concerned for water safety from the waste for the phosphate mines. Thank you for this. I've been looking for a place to understand water safety and don't know where to look. Happy to help, Tracy. So Mosaic, we know Mosaic has a major area, plays a major role in that area. Again, it's possibly a source of nutrients. I don't think it's ever explicitly been looked at as a source. The issue is when you're talking about marine algae, such as Crania brevis red tide, the limiting nutrient is nitrogen. It's not phosphorus. And so the extra phosphorus coming from Mosaic is not likely to be a contributor to the start of a bloom. Once it comes inshore, is Crania brevis using waste from Mosaic? No one, to my knowledge, no one's ever looked at that. So I can't give you a specific answer. All right, Stuart, confirmed. I've got your email. What is expected for the future with climate change? This is a way to end this on a Debbie Downer. So climate change, unfortunately, is expected to increase the frequency of harmful algal blooms. And it's been thought that the increasing in algal blooms that we've already seen globally can be linked to climate change and increases in temperature. Most algae species like warm waters, like warm temperatures. And so there is, you know, a link that, or a thought that the link in increasing in HABs is due to climate change, and that it's only expected to get worse. Climate change is not the only issue. Population increase is also a concern. Most people in the United States, Florida included, live on the coast. That's a huge, source of nutrients that we're contributing. It's more people causing more nutrients. And so we just have a lot of issues that we're gonna have to deal with and consider managing for blooms. And remember, red tide is a bloom that occurs naturally. And so we can only do our part to kind of make it less severe once it comes near the coast.
All right.
Okay, so, sorry.
Oh, go ahead. You will get into one more.
One last one. So what may trigger a Crania brevis bloom on the East Coast again? So that's ocean circulation. It's going to be whether or not the cells that are concentrated in the Gulf of Mexico get caught up in the loop current at a point in time when that loop current goes around the Florida Keys and is close enough to the East Coast where it'll bring it onshore.
All right. Thank you for that great presentation. That was very informative. And thank you everyone for joining. If we have follow-up stuff you want to send out to everyone, I can definitely do that. And then obviously we'll also post the recording on the blog, and also will be on the Facebook page if you want to watch it again. So
great. And I want to thank you all for answering my questions. It's really helpful.